fungi and microorganisms. And it looks a bit sensational. It reminds me of those old B-movie posters that you used to see when I was in adolescence and going to the movies on a Saturday for sixpence, for those who can remember sixpence. And you used to get the B-movie titles like Killer Bees and Killer Shark and Climbers Against the Killer Mountain. So when I saw this and it says Tree Killer on the Loose, I thought, oh, that's a bit sensationalist. But then I started delving into it, and actually, it's pretty sensational, and it's very appropriate. So the talk, I'm going to split it into two parts, because Phytophthora, there's still a lot of misconception about exactly what they are and what they're not. So hopefully the first part will sort of try and explain to you what they are and how they're classified now. And then originally I was going to talk about problem, but it's developed into actual problems. So let's kick off. Right, Phytophthora belong to Umycota, and the derivative of this comes from the Greek, two words, un and mykatos, which mean egg fungus, and it gets its name from the large female egg that develops containing female gametes. Now already we've got the word fungus in the title, and the other name is often used is water mold. And again, people associate molds with fungi. And a definition that's often used for Umycota is a distinct phylogenetic lineage of fungus-like eukaryotic microorganisms. Now, the problem is they're characterized by hyphal growth. And this is an example of what we call convergent evolution. For those of you who are not quite certain what convergent evolution is, it's essentially a process whereby organisms which are not closely related independently evolve similar traits as a result of having to adapt to environmental or ecological niches. And the classic example, which is always given, is the evolution of wings in insects, bats, birds, and going back as far as the dinosaurs, the pterosaurs. Now, they're all evolved wings, but by 4 independent mechanisms, but it's again a way of solving a problem. Now, the problem with this naming, it implies that Umycota are fungi, and they are not. They are clearly not fungi, and I'm going to show you why they're not fungi. Unfortunately, I reviewed 2 books this year, and both books referred to Phytophthora as being a fungus. And even the Forestry Commission, if you look down here, Phytophthora are a group of microscopic fungal pathogens. So there's a big problem in the terminology. I just want to show you a couple of slides that will show you the differences between Umycota and true fungi. And the near neighbours to these 2 groups are animals, and the nearest one to Umycota are diatoms and golden brown algae. As I said, they both got a hyphal type growth pattern, but in the Umycota, it's what we call coenocytic, and that means there's no division between the cells. It's essentially one long tube. They do have a slight division, but it occurs when they're forming spores. But fungi tend to have many divisions in their hyphae. They tend to be diploid, except for a haploid nuclei when they're developing gametes. But fungi tend to be typically haploid or dikaryotic. And this is interesting. The type of the genome, the size of the genome, is quite small in fungi, but these Umycota, they're actually larger, and they can go up to 250 megabases. Fungi, the main compound in their makeup, in their cell wall, is chitin, which is normally found in insects as well. But in Umycota, it's actually cellulose. So this is more plant origin than this, which is animal origin. They've both got these glucose polymers, and they're both 1,4, 1,3, and 1,6 linked glucans. But I've just recently found out that even though the side chains are the same, the enzymes are slightly different in how they're using in the enzymes that attach these side chains. Pigmentation, Umai are usually unpigmented. As we know with fungi, they normally have beautiful colours, especially if you think about the wax caps. At the moment, there's no secondary metabolites detected in Umai, but there's loads in fungi. You only have to think of the death cap. Mating hormones, they're non-peptide, but in this, they're usually small peptides. The asexual spore is undesiccated, which means it tends to have, it lives longer. Desiccated in fungi. This is an interesting one, and this is the one that gives their main classification. They're nearly all universal biflagellated. Sorry, I keep pressing the wrong button. We'll come back to that later. The sexual spores, oospores, are formed on termini of specialized hyphae. There's various types in fungi. You've seen examples of the large caps. The major energy resources are mycolaminin in Umai, but glycogen and associated sugars in fungi. If we look at the mitochondria, inside the mitochondria in fungi, they tend to be lamellar. In Umai, they are tubular. Now, when hyphae are growing in fungi, right at the terminal tip, you have something called the Spitzenkörper. And that is where you get the secretory vesicles and polar related proteins. And these are the ones that are normally associated with the tip of the fungal hyphae growing. Now, Umai also grow by extending the hyphal tip, but they don't have this Spitzenkörper. Augustosterol is the main component of fungal cell, but it's not found in Umai. It tends to be replaced by other phytosterols or cholesterol derivatives. Now, this has important implications because fungicides are traditionally being used to try and kill Umai. They have a limited success, but they don't kill. And also the lysine biosynthesis pathways, two totally different pathways. The DAP pathway is found in bacteria, algae, and Umai, but the AAA pathway is found in fungi. So where does it put it on the tree of life? This is one, you'll see many examples of these sort of diagrams in books and on the web. This is probably the best one and the easiest one I found to understand, even if it looks complicated. Down here we have the bacteria and also the archaea, which are not shown, but you've got this divergence from this common ancestor down here. But if we look at fungi, you can see how closely related to animals they are. If we go over to here, the brown algae and the diatoms, you can see Umai fit along with this class here. And they are as far away from fungi almost as it's possible to get. So again, this is one example of why people should treat them as non-fungi rather than fungi. This group here, the alveolata, the stramenopiles, and the rosaria, you might sometimes see as this SAR supercomplex. We'll come back to this in a bit, these stramenopiles, and show you how they get the name. Oh, sorry. My mouse has stopped working. They get the name stramenopiles from the flagella. Now, these are present during various cycle, and it always includes one Stramenopilus flagellum. And I'll show you a diagram of that in a minute to explain. Now, this is considered to be of such an important feature that the main author, Dick, who has written the definitive book on this kingdom, renamed them the Stramenopilia instead of Chromista. You might still see this name around, but this is the one that's now accepted as universal. And that derives from stramen for straw and pilus for hair. And if we look at the flagellum, the classic flagellum has what we call this 9+2 structure of microtubules. If you count them, there's 9 doublets here and a single doublet in the middle, and that's what we call 9+2. And in the classic flagellum, it's just this tubule that looks like this. There's a slight naked branch at the end, but this is often called the whiplash flagellum. Now, in the Straminopilis flagellum, this becomes coated in these hairs, and they're called tripartite tubular hairs. And this has often led to the name called tinsel flagellum. So Oomycetes have the whiplash and the tinsel flagellum, and this is one of the defining features, and that's what gives them the name Straminopiles. This is a lovely diagram I found off the internet, and it just shows you the zoospores, and they have 2 flagellum. And it's interesting that when they're in the developing sporangia form, when they're released, it's now been shown that they actually come out backwards. And these 2 flagella are actually downward pointing, and they come out backwards. But when they're outside, they then flip over and they flip the flagellum, so they're on the ventral aspect of the zoospores. And it's interesting that this one propels them forwards, but this one acts a bit like a rowing team, and it literally pulls them. So this one will drive them and this one pulls them. I don't want to concentrate too much on this because there's a lot of information here, but it's just to show you that Oomycota have been implicated in considerable disease mechanisms. Now, the number of species currently known is reckoned to be about 1,000, but I only looked up a couple of nights ago and they think it's 1,500 now. But as I'll show later, they think this is a chronic underestimation of the actual number of Oomycota, and it could be in the thousands and as many as 10,000. They are mainly saprotrophic, but they are extremely important in causing massive economic disease on animal, plant, and algae. These are just a few of them. We won't go over it, but just to show you that Plasmopara is an obligate one of the Impatiens genus, and it causes a downy mildew disease. And this is devastating plants in nurseries. One other thing that people also confuse is they think mildews are a single class. There are what we call downy mildews, which are caused by Oomycota, and there's powdery mildews, which are actually fungal in origin. Oomycota are involved in numerous problems in the fish industry, especially fish farming. And this one and this one cause various problems on fish species. Sorry, keeps jumping. So this is a problem in the fish industry, and it's now been estimated that it's causing problem across 4 continents, and about 20 countries are actually involved. This is an interesting one in that it's actually been found on mammals, especially dogs, but it actually has been found on humans as now. So, introduction to Oomycota and what they are and what they aren't. And now I'm going to concentrate on the talk on Phytophthora itself. The name is derived from the Greek, and it literally means plant destroyer. We think currently is about 180 species, and these are actually being named. But there's new species being described at an alarming rate. And again, towards the end of the talk, I'll show you how these are being discovered. Again, it's predicted the number could rise to at least 500 species, but again, that is considered an underestimation, and it could rise to 1,000. There's an increasing problem in that it's now realised that some of these species are actually cross-hybridising with each other. And one of the Phytophthora that's causing a huge problem with some trees are actually a hybridised example. And the trouble is, the hybridisation is producing very, very invasive species. In Britain, there's new species arising. These are some of the ones that I want to talk about. This is the one that probably some people have heard of, but these are new ones that have come in on the scene and probably are causing a lot more damage than the Remormon. Again, a bit like the fish industry, they're causing a significant economic and ecological losses. And 3 of these main pathogens were discovered in outbreaks in public parks and gardens. And this is an issue with biosecurity, with plants coming into this country. And what I didn't realise is that when a delivery of plants comes into Britain, it's only literally eyeballed. They just look to see if there are any symptoms. The trouble is, a lot of these plants and trees can be asymptomatic, and the problem can actually be in the roots, not in the foliage. So this eyeballing is a real problem at the moment. The Norwegians have taken a slightly different issue, and they're actually looking by a really nice technique to look for the presence of Phytophthora. I'll explain that a little bit more. The trouble is, with more plants coming into this country from other sources, and their ability to survive in soil, especially in the roots, we could have future invasions occurring. And as I said, this raises serious biosecurity issues. I know this looks a bit complicated, and it's a bit like the chicken and egg. Where do you actually start? Because it's a cycle. But if we look at the mycelium can actually go and produce sporangium at the tips, usually at the tips, and in the sporangium you get the individual zoospores being produced. Now these are motile, and then they can swim. They can't swim very far, and they don't survive that long, but they swim far enough to land on another host. But the sporangium sometimes doesn't develop these zoospores. It can actually land on a new host and produce what's called a germ tube. And this is effectively a new piece of hyphae, mycelium, and it can directly invade. The zoospores can actually fall onto the ground, and this is the way they can infect tubers in potatoes. Now, the infected plant then grows hyphae, and it starts all over again. Now, this is referred to as the asexual cycle, but this is the predominant one that occurs in nature, but every so often they will produce spores by the sexual phase. Now, in the sexual phase, it relies on hyphae or mycelium from 2 different mating species. When we think of fungal sex, we have to abandon all our conceptions of what we think of as the world, because in the animal world, there's obviously morphological differences, but in and fungi, what you have is compatibility systems. And what you're looking for is 2 hyphae of different compatibility that then come together. One will produce the male organ, and one will produce the female organ. You then get a meiosis occurring, where you get the haploid spores produced, the male ones and the female ones. You then get an oospore produced. Now that oospore then goes on to develop a sporangium, and the sporangium again can invade leaves directly by formation of a germ tube or by producing zoospores. So you've got the asexual phase and the sexual phase. When you produce these oospores, they're actually quite resistant and they can stop in the soil for, people reckon, up to 2 years. Now, what's not often shown in some diagrams is under conditions where the environment is not favourable for the Oomycete growth, they can actually produce what's called a chlamydospore. And again, it's an asexual organ, but this one has a very, very thick wall and is very resistant, and it can survive in the soil and in the environment for several years, perhaps even 8, 9, 10 years. So this one is a way that they can just lay dormant for years and years, and then suddenly the infection will come back. And again, they can produce sporangium and then produce zoospores. This is a lovely diagram, just shows you the sexual phase, and you get these two hyphae, a male and a female, and this just relies purely on genetics. There's no morphological differences between the two, and you can see the female forms an oogonium with the eggs inside. This forms an antheridium, and then you get this fertilization between the antheridium and the egg through this tube, and this will pass the gametes from the male into the female. Now, this is a beautiful diagram. This was only published very, very recently, and we could spend literally a talk just talking about this. But this shows you what happens when the zoospore or the sporangium produces a direct tube when it lands on a suitable surface. Now, the zoospores, when they land, they actually insist. They don't survive like this, but they insist, and then they put a tube out. And this is called the oppressorium. And then the oppressorium sticks to the leaf by adhesives. We're not quite sure what these are. And then it penetrates the cell. Now, it can penetrate the cell, or it can come in via the stomata. It was only 2 months ago where they worked out the mechanism of this, because in fungi, you get melanin produced and you get a rapid increase in water pressure at the terminal hyphae, which forces the hyphae through the cell. They couldn't quite work out how the Oomycetes works, but they've worked out that it almost works like a Japanese knife. Has anybody seen those Japanese knives that the sushi chefs use? They're called knifeu. And rather than working on a direct 90-degree angle, they work on an angle where literally you saw it through the cell layer. Now, what people don't realize is that plants have quite a sophisticated immunological system. It's not as complicated as a human, but it's fairly sophisticated. And when a pathogen invades, there's normally patterns, molecular patterns on the surface of these cells that are reckoned by pattern recognition receptors on the host. Now, this will trigger an immediate immune response. It's a quick response against an invading pathogen. This will trigger protein synthesis, will then trigger a response to produce effectors to try and combat the invading pathogen. One of the ones they produce is phytoalexins. Now, it's been shown that the Oomycota currently don't seem to have a defense against this. But what they do, and it's very similar to what happens in cancer patients who are known as they actually come inside, and there's a transporter system called ABC, which then pumps it back out. So these Oomycetes are very clever. And what they also do, the Oomycetes, they'll release compounds such as protease inhibitors. These are called crinkle type proteins, and these are RXL proteins, and they actually inhibit the production of the effectors that the cell is trying to produce to stop the invading pathogen. It's a bit of an arms battle between invader and host. Now, when this is in a semi-stable condition, this is described as biotrophic. It's almost like a parasitic relationship. This is trying to take as many nutrients out of the host as possible, and the host is trying to stop those nutrients being released. Now, we don't know when it happens, but sometimes this biotrophic relationship becomes what's known as necrotrophic, and it will actually then kill the plant outright. Whether it's triggered by it's exhausted the plant and there's no more nutrients coming out, but some Phytophthora will go straight for the necrotrophic mode and actually kill the plant outright. And then these produce spores that will drop straight into the soil. These ones can produce sporangia that come out of the stomata. But it's a fascinating system, and it's just a combat between the host and the pathogen. Now, how does Phytophthora cause damage? The infection results in necrosis, and a necrosis, what we mean by necrosis, is cell death. And it can be in the leaves, the stems, or the roots, or all three. If the infection's in the leaves, it's often referred to as blight. If it's on the stems or twigs, it creates what's called a canker, and they can leach resin out. They can be localized, or they can actually expand around the whole of the stem and effectively cut off the delivery of nutrients from the roots to the stem. And that can lead to a slow decline or a sudden death of the canopy. The Phytophthora can actually get into the water transport system, the xylem. And then if that gets in, you'll get an example of wilt. So we've all seen what happens when plants are derived of water. They literally wilt over. It can get into the phloem, so it disrupts the movement of nutrients, especially sugars. Many Phytophoras infect the roots, causing root rot. Now, some of them will only kill the fine roots. Sorry about this, but it's really playing up. And some get into the roots and then will actually progress all the way up the tree and actually into the root crown. As I said, you can get many symptoms or just a single symptom. Now, the problem is a lot of these symptoms are very similar to other pathogens, injuries, and what we call abiotic stresses. So that's where you get conditions where they get shortage of calcium, magnesium. Now, this should be familiar to a lot of people. It's the lateral flow test. And because a lot of these symptoms in the field are very similar to other pests and abiotic stress, it's important to get a rapid identification of the invading pathogen. And it's very similar to what we've been used to in the COVID. You take a sample, you put it into an extract fluid, you mix it up, you take a bit of sample, you put it into the slide, and then you get result that's either positive or negative. Now, this is only suitable for getting it down to genus level. So this will tell you if Phytophthora is present or not, but it won't actually tell you which Phytophthora it is. You then have to send it away to the lab, and they do what's called a PCR test. And again, some of you should be familiar with the PCR, the polymerase chain reaction. And what you do is you look for a specific amplified product that is indicative of each Phytophthora species. Now, the one most people are familiar with is potato famine. And this occurred in Ireland between 1845 and 1852. If you go to loads of statues around that sort of, right, but they highlight the problem that occurred in Ireland. This is one in Dublin, and you can see these examples of who are desperately malnourished. That about 1 million people died actually as a result of this disease in the country, but perhaps about 2 million actually fled the country. And the cause of the famine was a Phytophthora, and this is infestans. Now, I've actually made the classic mistake there of mentioning the word fungus. and it's confined to what we call solanaceous hosts. So that's tomato and potato. Now, people think it was just associated with Ireland, but it was actually found throughout Europe, and it caused about 100,000 deaths in Europe alone. Again, people think this is an old disease and it's gone away. It hasn't. It still causes problems worldwide and huge economic losses. And they reckon the global cost is estimated about $6 billion per year. And the loss in potatoes, they reckon, is sufficient to feed anywhere between 80 to many hundreds of millions of people. We don't know the true figure, but it's a high proportion. The genome was found to be considerably larger than most Phytophthora species. And they think this is one reason why it's still a problem, because in the genome, they found it contained a lot of what's called repetitive sequences, and also sequences that are called transposons. And this allows it to develop aggressive strains very, very quickly. And if you compare that size of the genome to other Phytophthora species, you can see the difference in the size. These are a few slides showing you the effect. It doesn't look too bad there. That's an early symptom of potato blight. Now, this is an interesting slide because one of the ways you can try to combat blight, apart from treatment, is to develop strains that are resistant. And this is a field of King Edward potatoes. This is a resistant strain, and this is one that's succumbed to blight. And you can see what happens. You have a massive devastation of the foliage. And if the foliage has gone, photosynthesis has gone, and then the production of nutrients and sugars for the plant doesn't happen and it dies. So this is an example of dieback. These are tubers from potatoes, and you can see you get a lot of this disease in a potato. Now, a lot of the rot that occurs in potatoes doesn't come from Phytophthora itself. The Phytophthora weakens the potato tubers, and then soft rot bacteria get in. And it's the soft rot bacteria that mainly cause the problem when they're stored. These are tomatoes, and you can see the effect on tomatoes. They're actually starting to die off. They think that the organism that caused potato blight originally came from Mexico. Now, potatoes originate in the Andes, but they actually think the infestation started off in Mexico because the potato came up through this channel, and there's a lot of mixing of cultivars around here, and they think it was the infestans that then arose. Sorry. And they've traced the first strain that arrived into Europe, and they think it originally arrived in Belgium. Then it was transferred from Belgium across Europe and into the UK. And they've named this strain uniquely Herb 1. But it looks like there was a second phase in the early 1900s, which they've now shown to be US 1. And this is completely different to the original strain. But as you can see, this has now spread back into South America, where the potato originated. It's gone into Africa, and it's gone all the way back into Europe and back across Asia. Now, as I said before, because of the genome, Dans, it can mutate very quickly and it can produce different. The strain that's causing the problem is the A2, 13A2. As I said before, for sexual phase to occur in Umycota, you have to have the A1 mating type and the A2 mating type. They can produce asexual spores by themselves, but when they come together, you get this sexual union, which produces a more diverse strain. But at the moment, it's the single A2, 13A2, that's causing a problem. And you can see it sort of started to arise perhaps 2004 and has now become dominant in 2008 and into 2010. And if you look at the map, it's in the UK, across all Wales, Ireland, England and Scotland. It's down in Europe. It's gone across Europe, and it's now on the borders of Europe and Asia. Not shown on this map is its problem in India. The A2 has caused, in certain areas in India, 100% destruction of tomatoes and potatoes in certain areas. So this is a very, very aggressive stain. And as I said, most people think potato blight has gone away and it's an old disease, but it's not. It's very much still there. Now, this is the one that's probably causing the most concern in this country at the moment. It was first found in the UK in 2002, and again, it was found in nurseries and in the horticultural trade. It infects shoots and leaves of ornamental shrubs, especially rhododendron, especially ponticum. And this now seems to be the problem, in that it is a host for Phytophthora ramorum that's allowing it to spread to susceptible trees, and it's the trees that are at risk. I know you get rhododendron dieback, but a lot of people would be quite happy if the rhododendron dies, but it's the problem with it's a host for ramorum. Now, typical symptoms include dieback and wilting, as we've seen. Oh dear, that's a large jump. So that's typical of the foliage, but you also get root rot. That's a normal root, and this is root rot. Now, the problem is, since 2009, ramorum has also been found sporulating on bilberry in heathland. So you now got the worry that you're going to have destruction of an ecological system based on bilberry. Now, ramorum, since the mid-1990s, was a cause of massive destruction of oak species in Oregon and California. And because it killed the oak, several species of oak, it was referred to as sudden oak death. And that's where we get the name from. At the moment, hopefully, and it shouldn't be, hopefully it shouldn't apply. It doesn't apply to the UK. The pathogen has been detected on oaks, but for some reason, the oaks are showing a resilience to developing the disease. The problem is it affects large trees and causes what we now call sudden larch death. There's about 3,000 hectares of larch in Wales, Devon, Cornwall, Somerset, and Northern Ireland, and thousands and thousands of trees have been felled. And probably the biggest felling operation at the moment is in Scotland, in southwest Scotland, in Dumfries and Galloway. The trouble is, this industry is worth £1 billion at the moment. Now, I've shown you that ramorum as a host on rhododendron, but it's now been shown that 150 plant species can possibly act as an host to ramorum. It does cause a problem in nurseries, and it's causing a problem in Washington and Europe. What's even more worrying is European sweet chestnut is also a host, and in southern England since 2015. Other species, again, which are worrying, are the fir trees, because again, they're part of the forest industry, and they can be infected when growing near infected larch. And recently, it's just been identified on Sitka, and Sitka is another commercial tree grown in this country. And you can see it's mainly down the western side of the UK. Southwest Wales is a big problem, but this is the huge problem. If you look at Wales, you can see in 2010, it started as a few outbreaks down here. And by 2018, it's now spread all the way up through Wales and across up into North Wales. As far as I know, Anglesea's free at the moment. They've split the Wales into 3 zones, and I'll explain a bit more about that when I show you the next slide. Because again, Scotland have had they've divided it into 3 zones themselves. It's a bit hard to explain, and it's a shame, but in Dumfries and Galloway, they've essentially said there's nothing we can do about it. The disease is there, it's that widespread, we've just got to live with it and manage it as best we can and fell the trees as and when. This is the zone that's causing the biggest problem, because you can see it's now spreading across Scotland, and this is similar to the 3 zones in Wales. And what they're doing is, if they find an outbreak in here, they're putting a plant notice out straight away for these trees to be felled, and trees within 250 meters radius to be felled as well. And this is hopefully a control measure to stop the spread occurring in here and don't come to the situation which we find in Dumfries and Galloway, which they basically said there's nothing we can do about it. This is a gross photo. You can see massive dieback occurring in a large plantation there. This is an example of what happens on the twigs and the stems. You get dieback on the foliage, and this is worrying because this is 5 miles from where I live, and this is in Bagger Woods in Barnsley, and you can see now the devastation that causes when you have to cull these trees. So luckily there's a few neighbouring trees which are of a different species which at the moment are infected. But if you go into Wales and Dumfries and Galloway, and unfortunately I had some pictures but I can't find them, there's just massive areas where the trees have been felled. Now another pathogen of Phytophthora is Kernovii, and this was first found in 2003 in a beech tree in Cornwall. Now again, at the same time, there was some scientists identified it on rhododendron, and they found it was the same organism. The scientist who discovered this lived in Cornwall, and he gave it the name Kernovii after the Cornish name for Kernow for Cornwall. It's now been called Kernovii, but since it was found in Cornwall, it's now been found elsewhere in Great Britain, mainly in southwest, but there are other locations in Scotland and Wales. The symptoms are very, very similar to remorum. The principal hosts are, again, rhododendron and again, bilberry. And these are worrying. They really are worrying with the bilberry. Hundreds of rhododendron plants have been heavily diseased and killed by it, as are areas of bilberry. Unfortunately, outside woodland, it can infect a wide range of ornamental plants. And people who visit amenity parks and woodlands and nurseries have to be aware that it's having a significant impact on these industries. Again, unfortunately, other trees and woodland plants known to be susceptible to Kernovii include beech, oak, laurel, holly, pine, and what's even worrying is the oak. Now, when a tree is suspected of showing an infection of remorum, it's actually tested for remorum and Kernovii, because it's important to know which one is actually causing the disease. Kernovii causes similar problems. Early stages, you get blight, and then you get massive dieback of bilberry. This is an example of the cankers. Now, this is the only map I could find. It's 2010. You can see it's mainly in the southwest, but it is showing evidence of spreading. And as far as I know it's now in the Cairngorms as well. This is causing serious problems. Now, this is one of the trees that is a hybrid. It was first discovered in 1993, but it's now considered widespread. And the total number of alder trees infected, they think, is at least a fifth. The highest incidence seems to be the southeast, but there's heavy losses occurring throughout the UK. It's also found in 11 European countries, including these. And a subspecies of this organism has even been found in Alaska, and another Phytophthora of this species has been found in Australia. It just doesn't infect one alder. It includes all the native common species and the other two species that are planted here. Other species in Europe are also susceptible. The only good thing you can say about this Phytophthora at the moment is it appears to just be highly specific to alder and doesn't seem to affect any other plant in any other genus. You can see here an example of dieback in alder. Now, alder is very valuable in stabilizing riparian systems, river systems. And if you get dieback of here, you get collapse of river banks. So you're going to get problems with flooding. Now, this is the one that's been shown to be a hybrid. And it's a hybrid between Cambivora and Fragaria, which is a pathogen of strawberry. And as I said, when you've got a hybrid, you can get different hybrids. And the problem is now, these hybrids are forming between various species, and it's spread across Europe as what we call the hybrid swarm. And some of them are very aggressive and very damaging. There's naming problems with it at the moment, but it's been agreed that the standard type of pathogen is Phytophthora olney, subspecies olney. And this is the most aggressive variant. There's different types, and they're known by different names. And the problem is, when we talk about hybridization, you can get hybridization between 2, 3, or 4 species. So you get diploid, triploid, and tetraploid species. The one we've got in this country at the moment is tetraploid. Another Phytophthora is Lateralis, and this is the main cause of death in cypress trees in North America. But in 2010, it was actually detected. Oh, sorry about this. In 2010, it was actually detected in a country park in Dunbartonshire. And the infection sites since then have been found in forest strands, parks, private gardens. Again, I hate to keep going on about this, but the most likely source seems to be plants coming into this country. And 4 outbreaks on mature trees in this country were all found next to garden centres or nursery sites. The outbreaks have been confirmed in southwest England, Yorkshire, Scotland, and Northern Ireland. But the disease at the moment is most prevalent on the western side of Scotland. And if we look at a global distribution of Lateralis, you can see where it's caused the death of the cypress trees. Again, in California and Oregon, they seem to be suffering as much as we are. There's a few cases in Southeast Asia, a case in Europe, but again, the UK seems to be a major source of infection. It affects Lawson trees, but again, look at its host range. Cedar, yew. As far as I know, it's not been found on our native yew. But if it's been found on Pacific yew, I don't see any reason why here. Other cypress species, juniper, which could be a huge problem, and it's been found in soil nurseries with marigolds and pomegranate. So again, these Phytophthora are quite promiscuous in the species they infect. Another one I want to talk about is Ostracedri. This was first found in Germany on juniper, and in 2007 it was discovered to be a cause of a massive dieback in cedar trees in Argentina, and it was there that it was fully characterized as Phytophthora ostracedri. In Scotland, again, it's been mainly found in amenity specimens, and it's been found in Iran. It affects juniper, and it causes dieback and tree death. And if we look at juniper, you can see the dieback that's occurring. Now, juniper is in a sorry state anyway, and it was already protected, and it was realised it was really vulnerable anyway, and that was before any Phytophthora infection occurred. And figures from Natural England have estimated that 35% of what we call SSIs in England are, about 66% of them are in unfavourable condition. They think in Scotland, the incidence of juniper that's in a real sorry state, irrelevant of Phytophthora, is about 70 to 80%. And since 1990 onwards, juniper has declined steadily, and it's important not only with game management but in nature conservation as a whole. Oh, sorry. And the problem is, with juniper being in such a sorry state, and if it's in a fragile state anyway, it's not going to have much resistance to Ostracedri getting in. An acceleration, and this could accelerate a massive decline in juniper. And you can see there, Cumbria has got a big infection in juniper, but in the Grampians and the Cairngorms, there is a huge infection of all the, well, not all the juniper, but the majority of the juniper in the Cairngorms. So you're going to get a massive destruction of the juniper ecosystem. Again, if we look at the map, this is in Argentina, and that's where it caused that massive dieback of cedars, but that's where it was characterized. One case in Iran. At the moment, the rest of the world is clear, but again, look at us. We've got an infection from a new Phytophthora species. The UK is not looking particularly good in a healthy state at the moment. I mean, we've got a problem anyway with climate change, ash dieback, elm effect, but this is having a huge effect on the trees. Pluviavis is another one, and this is affecting hemlock and fir trees. Again, it was reported in Oregon and California. It's been found in New Zealand, where it's responsible for red needlecast. But this is the problem. Only this year, September 2021, it was discovered in Cornwall on a mature hemlock and fir tree. I've highlighted this because this is the first actual report of this pathogen in Europe. So it's not anywhere else, it's just in the UK. Now, it's currently under investigation to see if this has just come in as what we call a passenger. So it's not actually causing the disease itself, but it's found there, or if it's actually caused the dieback of this hemlock and Douglas tree. We don't know at the moment because it was only found literally 2 months ago. This was a paper that I downloaded, and if you just look at the headline, Phytophthora cinnamoni, one of the world's worst plant pathogens. I mean, that's a good statement for a scientific paper. It's interesting that it's thought to have been in Britain for more than 100 years, and it seems to have been just lying there, not doing much. It's found on ornamentals, but now it's been found to such as sweet chestnut and the oak, and it attacks the roots and the root collar. It's causing ink disease in sweet chestnuts down in the southeast of England. The problem is it's distributed worldwide, and we don't actually know the actual number of hosts, but I'll show you a slide in a minute to highlight the problem. There's just a few there that I've highlighted to show you some of the hosts it on. It's very promiscuous, and it also found infecting avocado and pineapple crops at the moment. But current research has also shown that it can infect all these groups of organisms. So its host range is phenomenal. There's a huge problem in Western Australia at the moment, and they reckon 40% of all the species in Australia are susceptible. 14% are considered highly susceptible, and it's responsible, this organism is responsible for a massive dieback of eucalyptus trees. It's called the Jarrah dieback. And you can see there huge swathes of countryside in Australia that are showing dieback. Now, this is the problem. If you remember the distribution maps of some of the Phytophthora, they've been very localized. But if you look at cinnamomum, it's, I wouldn't say everywhere, but the distribution is absolutely incredible. Probably of what great concern there is Russia and China. It's a bit like their infection rates for COVID. We don't know the true rates of anything for Russia and China. And I've got a sneaky feeling they're hiding a huge problem, because there's the big forests in Siberia. I'm wondering if they are actually showing evidence of this, and the Russians are just not reporting it. But the current estimate for cinnamomum is 1,000 plus plants that can act as a host. So what's the future? Well, the future is metabarcoding. Now, this is a technique. It's very similar to environmental DNA analysis, but this identifies numerous species at one go. It relies on short transcribed sequences. So it's not the whole sequence of the organism. It's short transcribed specific sequences. And what you do is you get a barcode. But it's a good way of identifying numerous species at one go. And I've just put two studies here, and both were in Scotland. This is going on throughout the world, but these two are in Scotland. 2020, they found 23 species. Most of them are known, and these included 19 of 42 species recently listed as present, and 4 species not previously recorded. So what this metabarcoding does, it finds hidden Phytophthora that are just lying there, that at the moment are perhaps not causing a problem, but in the future could do. But this is interesting. There were 3 undescribed Phytophthora species and 9 Umycete sequences that have got no clear match to any known genus at the moment. And another study in 2019 did metabarcoding as well, and they looked at 14 amenity sites, and a high diversity was detected at all sites, corresponding to 23 Phytophthora species, as well as 12 undescribed Umycete sequences. So metabarcoding is the way forward. Now, I think we should be adopting this on our borders, because it's a quick, I wouldn't say simple technique, but it's a quick technique that can identify if a plant is harbouring any Phytophthora. And the Norwegians are actually doing this at the moment in their borders. As I said before, we're just doing a quick eyeball, which doesn't seem to be working. So there's just a final thought for you: a world without trees. And for those, it's just a lecture for people who have never seen a tree before. So I hope it never comes to that, but it's just a thought to leave you with. And thank you for listening.